Hi, my name is Daniel Troup. I'm a junior here at the University of Arizona studying physiology and molecular and cellular biology. As a member of the undergraduate biology research program, I work in Dr. Gandolfi's laboratory in the College of Pharmacy in the Department of Pharmacology and Toxicology with Tam Lee and Matthew Medeiros. In Dr. Gandolfi's laboratory, we study arsenic-induced bladder cancer. So why arsenic? Arsenic is a common natural contaminant found in drinking water throughout the world. Arsenic has been associated with increased risks of bladder, skin, and lung cancers. However, we and other researchers are still unsure of the exact mechanisms of arsenic-induced bladder cancer. So in our laboratory, we study a biotransformation of arsenic called Monomethylarsonous acid. We call it MMA3. MMA3 is 20 times more toxic than arsenic, and we believe it is the primary metabolite responsible for arsenic-induced cancer. The model we use in our laboratory to study arsenic-induced bladder cancer is an immortalized human urothelial cell line, which we call urotza. Urotza behave like transitional epithelial cells of the urinary bladder. So in our previous experiments, we have shown that 3 months of chronic low-dose exposure to MMA3 has caused irreversible malignant transformation of urotza cells with coinciding gene expression changes. So in our studies, low dose means 50 nanomolars or 3.75 parts per billion of arsenic, which is a relevant concentration as the EPA standard for arsenic concentration in the United States is 10 parts per billion. So at the time of 3 months of exposure, there has been significant gene expression and phenotypic alterations, including increased cell proliferation rate, increased cell damage, increased cell size, generation of reactive oxygen species, growth in colony formation in soft agar, which is a representation of epithelial to mesenchymal transition, a characteristic of cancer, as well as formation of tumors in severely combined immunodeficient mice. To study these transcriptional changes, we analyzed by Western blot analysis the protein expression of 4 biomarker proteins associated with bladder carcinogenesis, whose gene expressions were significantly altered following 3 months of exposure from MMA3. These proteins were STAT3, TMPRSS11A, MKP1, and IRF1. In our current study, we demonstrated the validation of MMA3-induced transcriptional changes in urotza for MKP1 and IRF1 as their protein expression was decreased in a time-dependent fashion just like their gene expression following 3 months of exposure to MMA3. As for TMPRSS11A, its protein expression did not yield any significant results, and for STAT3, its protein expression increased through 3 months, contradictory to its gene expression, leading us to believe that there may be some post-transcriptional regulation going on or misleading results from our gene array study. So with this validation of transcriptional changes induced by MMA3, we can further investigate possible molecular targets in urotza of MMA3, anywhere from cytosolic proteins to genetic factors or epigenetic factors that MMA3 interacts with, causing these alterations and subsequent cancer. So other future studies in our lab involve finding possible repair mechanisms within urotza to fight off damage from MMA3 exposure within the first 2 months. Finding these cellular mechanisms will help us lead to insights about how the cell doesn't transform until the 3rd month of exposure. So as a member of the undergraduate biology research program here at the University of Arizona, I encourage all of you, whatever school you're into, to get involved in research if there's programs at your school. Research is fascinating. You're always busy. You always find yourself involved in what you're doing, and it always relates to what you're studying in class. And a career in research is fantastic because you will always find work. Once you find something on research, it never ends. There's always something new to study, so you will always have a job. So I persuade you to look into the field of research. Thank you.